Hi, greetings everyone. This is Matthew Cherian from United States. I'll be chairing this session or the webinar, and my co-pan moderator, Nicolene Abiskik, will be joining us shortly. Today's panelist is Dr. Jaspreet Arora. He is a PhD in chemical engineering with very broad experience in multiple aspects of drug development. He specialized in dispersed dosage forms like liposomes, polymeric nanoparticles, and all other related areas. Dr. Arora had been in the industry for over 10 years, holding leadership positions in research and development and also in business development. He will present on multi-particulate technology for pediatric dosage forms. So you will have an opportunity to ask questions of Dr. Arora, and it is done by going to the bottom of your screen. There is a Q&A portal. You can click on that and type in your questions. We'll take your questions at the end of the presentation, and Dr. Arora will answer them one by one. So over to you, Jaspreet.
Sorry, I had to unmute myself.
Yes.
Hello, everyone. Try sharing my screen again.
Please.
Okay, hope you can see the screen. Good afternoon, good evening, good morning, wherever you are in the world. Thank you so much for joining this FIP event. Thank you, Dr. Cherian, for introducing me and giving me the opportunity to present in this webinar. So we will be talking about using multi-particulate technology with its focus and applications in pediatric drug products. So just an overview of the agenda for today's talk. We would want to understand, we will look at what are the different considerations for pediatric dosage forms. What factors do you have to think of when you are working on one? Then we will go into introduction to different types of multi-particulates. And after that, we will try and understand what use of a couple of technologies based on 2 case studies. And then we will have time for questions and answers. So why is there a need to even talk about pediatric or geriatric or patient-specific drug product technologies? Most of the drugs that are made for the adult population. And there can be indications or where the adult patients are suffering for one indication. But a lot of times, in a lot of cases, in millions of patients, the adult dosage form is not suitable for them. For instance, for pediatrics, which we'll see in the next slide, you cannot give a decent sized tablet because the infants or neonates especially, or even toddlers, will not gulp or swallow a tablet. There are some geriatric indications where there could be comorbidities, where it makes it difficult for just focusing on that indication and not thinking about what else the patient is going through, whether in terms of other medications, whether in terms of the physical attributes that the patient is going through, because they are in a certain indication or disease. So that all has to be thought of. Dr. Sven Stegemann from the University of Graz in Austria has published this paper, and he has published multiple papers talking about patient-centric dosage design. So if you'd like to learn more, please follow Dr. Sven Stegemann's literature. So one of the big things I was talking about, which is pertinent to pediatric formulations, it's the swallowing. As you can see in this table, it takes about 2 to 3 years for children, for neonates, infants, to be able to swallow. Neonates and infants and toddlers do not have that ability. So if you want to treat them for a certain indication or disease, it's going to be very difficult or pretty much impractical to have a tablet form of the dosage form. And so that is where you have to think out of the box. You have to think outside of the conventional dosage forms, such as tablets that are available for drug delivery. So if you look at the different options that you have that are approaches to pediatric populations, you have to think between industrial requirements and patient needs. What I mean by that, the industrial requirements may need you to be able to manufacture these with a quality and precision, batch to batch. But then you also have to think of what the patient needs are. Whether do you need a dosage form that can have a flexible dosing regimen, whether you can deliver multiple doses with the same drug product intermediate, how much stability or transportation issues will you need? Are you going to be going to different climatic zones? So all these things. Taste masking is also big in multi-particulates for pediatrics, especially in the adolescence range where they are taking some kind of an oral formulation. Um, so all these things are something that one must think about when they're talking about pediatric formulation design. And if you look at the options listed on the slide, multi-particulates tick most of the boxes compared to chewable tablets, liquids, or powders that are reconstituted in the media, and then or oral dispersible tablets or films. They all have their pros and cons, but if you design multi-particulate technology platform properly, you can circumvent most of the issues and check most of the boxes that are needed. Um, this is the other advantage about multi-particulates, thinking about the concept of universal design. So if you are a sponsor trying to create a drug product, let's say even for adult population, but then you need something down the lane going from immediate to modified release, or you need to have your pediatric plan, you can think about multi-particulates if it fits in your target product profile, which we'll talk in the next slide. But multi-particulates enable you to have a universal design, meaning it could be applicable to all patient populations, including adults, pediatrics, and geriatrics. You have to know what are the key development, manufacturing, and patient requirements for you to be able to make work with a dosage form in the multi-particulate platform family. So we have talked about multi-particulates and how they are great for pediatrics. What are they? Let's see what it is. So as the name suggests, multi-particulates are all about breaking the monolith. Monolith being you have a tablet, just imagine cutting that tablet into many, many small pieces. And doing so, it gives you advantages and make them amenable to excuse me, other processes, unit operations that are harder to do with tablets. Um, and you can divide the dose in multiple discrete units that are easily swallowable compared to a monolith. And these basic advantages and just breaking the monoliths opens up a lot of applications and a lot of dosage design advantages that a tablet or a monolith unfortunately does not have. Just to show you commercial precedents for multi-particulates, there are quite a few commercial products out there where the final dosage form is flexible. You can find suspensions, you can find sprinkle caps, you can encapsulate one type of bead or multiple type of beads in capsules. You can use capsules with multi-particulates for fixed dose combinations. You also have orally disintegrated tablets. You can also have mixed salts in tablets. And one of the advantages is you can, with the same drug product intermediate, or if you make a certain bead, you can use that and fill that with a different weight and hence different dose to get, to different strengths of or dose of the same medicine. So it really is a very versatile technology platform wherein the final dosage form is flexible and you can decide that based on the target product profile. Um, Other than some of these considerations, there are drivers for multi-particulates that I think we should cover. One of the differences in how the body handles a monolith tablet versus multi-particulates. So let's say you take some beads, you sprinkle them with food, or you have a suspension, the patient drinks it, or you just take a capsule with the capsule gel quickly dissolves, and now you have all these beads in your stomach. One of the biggest advantages of multiparticulates is that the stomach emptying time, also referred to as the gastric residence time, is short for these in the fasted state, usually between 30 minutes to an hour. And there's less variability. If you look at a tablet, on the other hand, you need the tablet to, if it is bigger than 7 or so millimeters, which is the pylorus rectum, the opening that connects the stomach to the intestine, you need the tablet to disintegrate. And then the granules or the drugs is dissolving in your gastric environment is then going to find its way to the intestinal parts and then be absorbed. But there's inherent variability in the gastric residence. Multi-particulates help circumvent that because they just pass through that opening and go straight from your stomach to your intestines. And because of that, you get less variability, and less variability gives you an improved IVIVC, in vivo, in vitro correlations. Multi-particulates can also be engineered to eliminate dose dumping concerns. Going back to thinking about the patient, there are some indications where there is a potential for abuse, and also there's a potential for alcohol dose dumping. These concerns can be eliminated by having the right coatings on multi-particulates, which do not dissolve in presence of a lot of alcohol. So that's another area where multi-particulates are used. And then one of the things that we will talk about is you can tailor the dissolution profiles. You can obviously have immediate release multi-particulates, but you can have modified, delayed, sustained release multi-particulates, and we'll look at one of those case studies. One of the very good things about multi-particulates is that their size is small. And if it's small enough, you get an acceptable mouthfeel. So the mouthfeel is also an important aspect when you're thinking about pediatric formulations. And some of these multi-particulates, like lipid multi-particulates, have some taste masking baked into them. We already talked about the flexibility in dosage form, the final dosage form, and then reducing intervariability. Apart from these technical advantages, there are also some economic drivers. The most common one is the 505(b)(2) pathways. Some of our regulatory colleagues on this webinar here will recognize that the EMA and the FDA have motivated sponsors, innovators to design and implement pediatric trials so that they can extend their exclusivity even in the adult population by 6 months or so. And that added exclusivity or that economic driver leads to them motivates them to develop these drugs for pediatric population, where in some indications the pediatric populations could be ignored just because it is not trivial economically, it's expensive, and then they just don't have that much patient pool or the business case is not there. But this actually helps sponsors proactively develop their pediatric plans and dosage forms. The other thing that's pretty big is the patent extensions. Apart from the pediatric exclusivity, when it comes to life cycle management, when you're going from, let's say, once a day dosing you're going from twice a day dosing, BID or TID, to QD, once a day dosing, you can engineer multi-particulates for modified release so that your blood plasma concentrations are above a threshold that gives you that therapeutic effect in the patient population that you're looking for. So not only they're technical, but also they're economic drivers for using multi-particulates. All right, I have mentioned this in a couple of slides. Target product profile. What does it mean? So when you decide to work in an indication or a disease area, and you're thinking about how you're going to develop the drug, you have to think all the way down the end. You have to be able to be on a path that shows you the path at the end of the tunnel. What I mean by that is if you are um, formulating a drug product for patients that are pediatrics, you know that you need some kind of a multi-particulate formulation, if especially if they're younger kids that you're looking forward to. What are the other patient-specific requirements that you need to be thinking right away? There are also performance and safety aspects. You have a target release profile, whether it's immediate release or modified release. You need the drug to be sufficiently absorbed. And, for some of those cases, you need bioavailability enhancement, and you have to select the right excipient. So that's the performance and safety aspect. There are the patient centricity and what are the patient needs, but also what is the final dosage form? Is it going to be a sprinkle cap? Is it going to be multi-particulates filled in a capsule? What what kind of scalability, packaging, all these considerations make up what's called a target product profile. So you put a lot of thought initially, so that when you see what is intersected by all these three different circles, you are able to find the right solution that has a path that can go all the way to commercialization while getting you to therapeutic effect that you need in specifically the patient population that you're targeting. So thinking about the target product profile is very critical from the very initial get-go. And you have to figure out what are the different stakeholders and quality, your patients, your dosage form, manufacturability, and then you build a roadmap or a framework of technology selection, and then figuring out what is the solution that fits your drug product development needs. All right, so we have looked at a few multi-particulates. We have defined them. Let's see what different type of multi-particulate technologies are present. So the first of, and this is something that I've worked on for years, is lipid multi-particulates made from the melt spray congeal process. Most of these multi-particulates that you see here have commercial precedence. So as you can see, based on the table, the different multi-particulates are um, differentiated by the processes that they're made from, the particle sizes that you get, and the typical drug loadings that you can achieve. Some are narrower than the others. But lipid multi-particulates, and we'll get into the details of this with a case study, have a lipid waxy core, which can carry crystalline or amorphous drug. Then some of the more common multi-particulates that you see are spray layer dispersions or beads, where you take a core and then you spray a layer of active drug and also different functional coatings that you can layer on here. Then you have pellets, which can be made with the CPS pellets or wet granulation or spheronization process, wherein you can try to incorporate the drug within the core. So you don't have a neutral core, you have an active filled core as well. And then mini tabs, which is also very interesting. It's basically akin to tablets, but they're much smaller form. You can go from 1.5 to 3, and in some cases, you can go down to about 1.1, 1.2 millimeters. And mini tablets also fall into that category where they can pass through the opening between the stomach and the intestine, therefore having reduced gastric emptying and less variability. So you get that benefit too. And in some of the cases where there is an adult formulation for tableting, if it has manufacturability in being a mini tab, that is often one path that sponsors like to take, where they already have spent the CMC work upfront to work on a tablet formulation, they can translate that in most cases, in some cases, in a mini tab form of the drug. The case studies that we will focus on this talk will be one will be with lipid multiparticulates and one will be with spray layer dispersions. So, all right, you have a few different types of multiparticulate technologies. So the first choice that you have to make is, what is the base model that I'm going to choose? You look down, you think of different buckets. What is your target release? Is it immediate or is it modified? That's one of the first decisions. And then the other thing is, the drug form based on the physical chemical properties and the BCS classification of the drug. You need to figure out whether you will be able to deliver a crystalline form of the drug, which would be able to dissolve and being absorbed sufficiently, or you would need some kind of solubility enhancement. And as you can see, if you need, let's say, both solubility enhancement and modified release, your options kind of trim down. Although now there are ways you could do a little bit of solubility enhancement with a lipid-based LMP as well. But let's not get into that. But the point I'm trying to make is, again, going back to the target product profile, what is your release target? What is the form of the drug that you're delivering? That helps you select the base model for the multi-particulate. And then sometimes the base model is not enough, which is absolutely okay. This is where the advantage of you can now coat multi-particulates with various coatings, whether it's an enteric coating where you're trying to prevent release in the acidic stomach environment, or whether it's a cosmetic coating where you want to differentiate your beads in terms of appearance and sometimes even patient compliance. Yes, colors do matter. That's why you have commercial products that are colored. Extended release, or that's where you can try and target a particular area of the GI tract where you want release. Also, time target release falls in that same bucket. Or you can just have different pH targeted release coatings, which above a certain pH, they will let the drug release to happen and the dissolution will commence. So, and a lot of times, you could have the base model and then you figure out what your needs are for the dosage form, and then you can add on multiple coatings on multi-particulates. Okay, so looks like we're doing okay with time. We can spend a little bit of time diving deep into the two case studies. The first case study is on coated lipid multi-particulates. Here, goal was to deliver modified release products, but create 3 different release profiles, which would hopefully have 3 different PK profiles in vivo in human patients. And it was a pediatric patient population, and the drug was sufficiently soluble to enable crystalline form use. And then because of the other factors considered in the TPP, we went with a lipid multi-particulates. It had a relatively low drug loading, which could be challenging when it comes to having a controlled release matrix, which we will look at. And then we had to put an enteric coating on the lipid multi-particulate because the drug had a pH-dependent solubility. If the drug started to dissolve in the acidic stomach environment, we would have gotten a burst or an immediate release effect. So to help prevent that, we had to coat the LMP with an enteric coating. The dosage form initially was just loose powder, and that was so that there is in the initial clinical studies for dose range finding in animals or looking at ascending dose studies, it is easy to just fill more at the site for the initial phase 1 human studies. And the target site that we were looking at is the upper GI tract. So yes, we want some kind of a modified release, but we don't want it to be released over a period of 12 to 14 hours. We want to focus on the upper GI tract. So we were looking at a release window of 4 to 6 hours. And then the particle size had to be less than 400 microns. It ended up being much less than that, as you can see in this SCM image. And the reason for that is if you have particles that are less than 300 microns, you have an acceptable mouthfeel, and you do not get that gritty taste feel in your mouth, which is important for the pediatric formulation. And as any solid oral dosage form, stability is a big part of it. So you shoot for at least 2 years stability at room temperature. So this was the target that we were working with. A little bit about lipid multi-particulate technology. As you can see in this schematic here and this image here, so the LMP process, LMPs are made from the melt spray congeal process. As the name suggests, you take the lipids, you melt them. You add the crystalline API and other excipients that are needed for a controlled release formulation. The ratio of your lipid matrix, and which has a certain permeability, which you can then play with by adding a pore former. So the ratio of the pore formers to the lipid matrix is a handle for getting faster or slower release as needed. So you have your excipients, your API, your lipids, they're molten in a wax. If it's crystalline API, it's not melting in the lipid melting temperature. It's going to be a suspension. And then you take that suspension and you shoot it in its liquid molten form over a very rapidly spinning disk. And as the lipid gets on the disk, it spreads on the disks, and then it's coming off, you have these beautiful ligaments that are formed that eventually shoot off as drops, and these drops quickly congeal and they are collected in a bag. So obviously, one of the initial requirements for a lipid to be manufacturable using this technology is that it needs to rapidly congeal. If it does not rapidly congeal, you cannot make them using the MSC process. Luckily, there are a few matrices that are available that can rapidly congeal, and hence, there's also commercial precedence for this. But this is how LMPs are manufactured. Because of this simple formulation space where you have one lipid matrix, which is the carrier, and then you have pore former, as needed to tune your release profile, you can get up to 60% of drug loading, and you can also work with 5% drug loading. So sometimes working with much smaller drug loadings can be a challenge because of uniformity issues, which we'll see in the next case study. But those could be circumvented again by just going to the multi-particulate form. All right. So technology selection. So once you've selected the lipid multi-particulate, you need to figure out what your formulation is going to be. So you have a target release rate, you know your API's physical chemical properties, and then from there, if you have formulation map, you can understand if you need a high permeability matrix for immediate release, or a low permeability matrix, or if it's just too slow that you need to have multiple coatings on top to get you the release profile that we need. So if you see in this in vitro dissolution curve, there is a gap here. This is the enteric coating. That means there's no release in the stomach. And then as that enteric coating dissolves, once it hits the close to neutral pH of the intestine, you have 3 distinct releases, which is based on a low permeability matrix. So this was the groundwork in helping us figure out what is a good starting place for the formulation. Now, this is where things get very interesting. Some lipid multiparticulates made with lipids that are amenable to polymorphic changes need to undergo another unit operation, what's called annealing. This is very interesting to me because when I was focused on this work, it got me to read a lot of chocolate crystallization, because when you're manufacturing chocolates, you have these lipids which have the same alpha to beta polymorphs, and you have to anneal them to get to the right taste, which is based on the right polymorph mix. So it was a very interesting digression tangent that I took, but helped me bring those learnings back to in annealing and figuring out when annealing is complete for lipid multiparticulates. So remember, we were talking about you take a melt, you have these ligaments, and you form these drops of liquid melt lipid, and then it quickly congeals. So it turns out that some of these lipids that can be a mixture of mono, di, triglycerides, when you quickly congeal them, they go to the alpha polymorph, which is the less thermodynamically stable polymorph, a high energy polymorph. And over time, it would want to morph into or transition to the more stable beta polymorph. And this is what annealing is centered around. It's basically you get these quickly congealed lipid multiparticulates. If you do a dissolution run, you will see that they will have a certain profile. But then if you just put them on stability, whether it's a long-term stability condition like 25 degrees Celsius, 60% relative humidity, or accelerated 40 degrees Celsius, 75% relative humidity, you will see that there will be changes in the release profiles. And that is something that obviously you do not want. Once you get a certain dissolution curve, you want the release profile to not change. So this is where annealing comes in, is it is the inherent potential of some lipid multi-particulate formulations to, right after they're made, stay in their high energy polymorph, and they have a potential to go to their more stable polymorph. And what we're doing with annealing is just making that fast. So something that might take a year at 2560 could take a few weeks at slightly elevated temperatures and humidity. So this is where the study of annealing comes in. Just to give you an example of how annealing can change different characteristics, these are placebo lipid multi-particulates. That means they do not have any active in them. So it is just the lipid matrix and a pore former, and which is also referred as the dissolution enhancer. As you can see in the SCM, that based on the degree of pore former and the degree of time, the amount of time that you anneal at accelerated conditions, you start to see these surface blooms. So you definitely see there are changes happening on the surface, and there are changes happening within the particle, which are important to understand. Another interesting experiment that was done is we fluorescently labeled the dissolution enhancer, and then we added it to the lipid matrix and made films of it. And initially, when the films are cast, you can see that the dissolution enhancer is pretty homogeneously distributed throughout the film. But as soon as you anneal the film, you see that there is migration, there are changes. So that tells you that the effective permeability of these films, and hence the spherical form of them, will change. And when the permeability changes, you do get a change in the release rate. Now, one thing that happens as you go from the alpha to the beta polymorph is the more stable polymorph is denser. So if within a fixed volume of a particle, because the volume and size of the particle is not changing, but within that particle, you're getting densification. And because of that, the effective porosity and hence permeability of the particle will increase to a certain degree. Another endpoint of annealing is looking at its thermal properties. So when you take a lipid and you run a DSC, which is differential scanning calorimetry scan of it, you can measure at what is the melting temperature and the melting enthalpy of the peak. And this is interesting because if you were to design a typical annealing study, what you would have to do is you would have to make these LMPs and then put them at different accelerated conditions, let's say 35, 60, 40, 75, 50, 75, maybe even 60 degrees. And then it is akin to doing what's called a ASAP style temperature study to look at physical or chemical degradation. So it's akin to that. But then what you have to do is, because dissolution is the final endpoint, you have to take these beads at different times, incubated at different temperatures and humidities, and do the dissolution. Now, the dissolution run for modified release can be up to a day sometimes. So that's a lot of work. So one of the things that we were thinking about is what is the other way that can indicate that annealing is complete? So if your dissolution profile keeps changing, and then when it stops changing, then yes, annealing is complete. That means now these release profiles would not further change, and whatever stabilization of the polymorph was supposed to happen has already occurred. But it is time consuming to keep running this solution on so many experiments. So going back to the thermal properties, that also can be an indicator when you're following the melting point, because you will not get a clear melting point. It's because most of these lipids are mixtures of monodiatribe, but looking at the maxima and see how it shifts, and looking at the enthalpies as they increase, because a more stable polymorph, like beta polymorph, will have a higher enthalpy of fusion. So you can track these changes and try to, once you have the data packet, you can use thermal markers basically as an indicator of the completion of annealing, and then you can do your confirmatory dissolution tests to make sure that there is sufficient annealing and you can progress packaging your multi-particulates. This is where you see that as you keep annealing these multi-particulates, the dissolution of these get faster and faster depending on some formulations. And obviously the dissolution kinetics differ as you increase the pore former or dissolution enhancer ratio in the formulation. So that acts as a handle as well. So just to briefly summarize this case study 1, we see that in controlled release lipid multiparticulates where you have a lipid choice excipient that is amenable to annealing, which means upon rapid congealing, it goes to the less stable high energy polymorph, you have to think of annealing as a way to stabilize the release profile. The drug release is proportional to the end release of the dissolution enhancer. And that is something that you can see through FTIR. You can look at polymorphic changes through DSC. You can look at the thermal markers for completion of annealing. Confocal showed you phase separation, and SEM showed you blooming. All these changes, the physico-thermal changes that are happening, basically increasing the permeability in the specific case, which led to a faster release. So all this analytical characterization made us understand what are the physical changes, what are the thermal changes that are thermodynamic changes that are happening, and that helps us understand what we're dealing with. So yeah, just to summarize the case study, you can see that lipid multiparticulates, you can have a tunable release based on the ratio of your lipid matrix using a high or a low permeability matrix coupled with a dissolution enhancer can get you very varied dissolution rates. Some excipients are prone to polymorphism and phase separation, which need to be annealed to achieve a stable shelf life. And how you get there is elevated temperature and humidity conditions. And a dissolution definitely is the benchmark marker for completion of annealing, but there are other properties that you can look at based on the polymorphic changes that can help you maybe less dissolution tests to figure out when annealing is complete. Okay, now we can jump on to our second case study, which is on spray layer dispersions. So this is another pediatric formulation where the goal was to deliver a very low dose of a drug using particulates to enable versatile dosing options for clinical trials and also the pediatric plan. Here, in this particular case, the drug needed to be amorphous just for uniformity. It was not that it was a low soluble compound or solubility was low. It was just that when you change the drug to its amorphous form, the dissolution of it is there's less variation in that, and also the distribution of it can be more. You can imagine if you have a very low amount of drug in a film around a bead, and if you have discrete crystalline particles, uniformity can be an issue. But in this particular case, if you dissolve the drug and you spray it with a polymer, it spreads evenly around the polymer coating onto beads of your choice. So that helps with uniformity. So sometimes the amorphous form is not needed for, because of low solubility, it is needed for uniformity. Obviously, it's a very peculiar case for that. The other challenging part was the drug loading in the particles was less than 1%. And there you get the challenge of, well, if you're going to fill this in capsules, you need a uniform dose. And since it was a spray layered bead, which was getting into a capsule with immediate release profile, when we were doing the initial risk assessment, we figured out that the uniformity and assay has to be de-risked so that it has a scalable and manufacturable path to commercialization. So just a bit of introduction to spray layer dispersions. Spray layer dispersion is when you take a sugar-based cord and you have a layer of polymer. Now, this polymer can have drug in it, which can be either crystalline, that is, if you, let's say you have a water insoluble drug and you're using an aqueous solution, you can have the crystalline form. Or sometimes you use a mixture of institute and organic solvents to dissolve the API to layer the amorphous form of the drug onto these beads. And these are usually done in a fluid bed coating, and the schematic you see here is this Worcester coating from the bottom spray, which is a very precedented and scalable technique to create spray layer dispersions. The problem statement in this particular case was that A, you have a very low dose, B, you have a small particle size. Now, with that, the sugar core that we chose, larger particle size, has less friability, just because we needed the acceptable mouthfeel as well. We needed a small particle size, and the problem with the small particle size is substrate friability. So there were definitely changes in the process that had to be very slow, long coding to get to that. But to mitigate the issue of substrate friability, which in then changes your content uniformity issues, we decided that we have to develop a PAT tool, which is process at line technology tool, which can help us predict solution endpoint, which will help reduce variability in the substrate batch circumvent the variability in the substrate batch to batch friability, because these different batches of just the excipient of the small size sugar core had different friability. So this is where we had to get creative, and then we had to follow a risk-based approach. And through that approach, we decided we need a PAT tool that's going to help during scale up, tech transfer, etc. Another reason what we wanted to do this as part of a risk assessment is that when you are coding and you do not have a way to determine a solution endpoint, which means I have X kilos of solution to spray, and if I spray X kilos, I am hoping to get 100% label claim. If you go with an assumed efficiency, which means that there is a spray efficiency in the process, which has various variables, including substrate friability. If you assume a spray efficiency and you have differences in friability, you will see that your potency starts to vary very quickly. This plot was made using the JMP software, which is a powerful tool for such kind of visualizations and very easy to do so. So this was another reason that we needed to develop a PAT tool to help circumvent that. Not spending too much time on the method itself, but the idea was that we developed a tool where you would take the sample that's being interim sample from the fluid bed, so the partially coated particle, you quickly dissolve it, you separate the API, you run a quick UV assay, and using the software, we use the Mettler-Toledo UV7 device, which has the LabX software, which you can program to do quick calculations as well. And the great thing about this method is we did not need an IPC person or an analytical person in the manufacturing suite. Yes, the operators themselves were able to carry this because the design was simplistic. All the operators did was they took a few beads, dissolved it in a solvent that just dissolves the API, but makes the polymer permeable enough to let the API dissolve, quickly filter it, run a UV, and the software tells you, okay, on this particular day, this is the particular spray efficiency, this is the amount of spray solution that you have to spray to get close to your percent label claim. And this helped reduce sensitivity to deviations in the materials. But then when you saw it in action, because of the robust and simplistic design, when we were doing our operator training, we looked at the data the data generated by scientists. These are analytical chemists that have years of bench experience, and the operators. And you can see that the numbers, the percent RSD is not too different. And then the actual magic was seen when you successfully made clinical batches across different scales, where if you use this method, your potency has a very narrow RSD. But if you, from the previous batches experience, if you assumed an endpoint, there's a lot of variation in the potency that you get. So developing this PAT tool based on a risk-based approach was very critical in helping us prevent the substrate friability issue and its effect on potency. So to quickly conclude case study B, this is where when you're thinking about developing a multi-particulate or any drug product, you need to think of all the challenges that you can get. So that's your target product profile. And then looking at issues based on the choice of technology. So in this case, we had particular sugar spheres that were small in particle size that were prone to variation in friability batch to batch, which could lead to different potencies of the batches. We had to de-risk that using a PAT tool. And the execution of that is also very important. Sometimes you can find solutions which are a little complicated. The execution of that is equivalently important because sometimes the manufacturing suites are not big. So you cannot have an IPC or analytical person running there. So we designed this in such a way that the operators themselves could execute them. And their percent error in executing this analytical test was equivalent to experienced bench analytical tests. So this is where I will pause. Thank you so much for your attention, and I think we are ready for questions. I will stop sharing my screen.
Thank you very much for your insightful presentation. So during the presentation, we collected some questions, so I will be reading them out. So what factors usually drive the decision between using mini tablets versus pellets in a pediatric formulation? Is it more about manufacturing feasibility or patient acceptability?
So that's a good question. So I think you have to think of all these things. You have to think of manufacturability. Now, both mini tabs and your pellets, whatever type of multi-particulate technology, pellet technology, are scalable as well. But sometimes what happens is the tablet formulation designed for adults is not amenable as a manufacturable, scalable formulation for mini tabs. So that's something that you have to keep in mind. Sometimes the patient populations like neonates, you need something to sprinkle on food or in their milk formula. So you don't need a slightly bigger version. So you have to go with multi-particulates. So yes, you have to think of the patient population, the patient needs, but also not only manufacturability, but if there is something that you're starting from, like let's say transitioning from an adult tablet formulation to a mini tab formulation, you have to see if that formulation is scalable in the mini tab form.
Great, thank you. We also had a few other questions. What is the advantage of NP formulation over traditional tablet or liquid preparation?
Yep. So we talked about the advantages of multi-particulates over tablets. So going from a monolith, which is a tablet, to breaking it down in discrete units with doses brings you a lot of advantages as multi-particulates. It makes you amenable to flexible dosing formula ways. You can put them in capsules, you can put them in sprinkle caps, you can put them in sachets, or sometimes you can compress them in tablets. That's not the point here, but it gives you flexibility in dosing. It opens you up to different patient populations, such as pediatrics and geriatrics, where they have problems swallowing, where tablets will not work. So that's on tablets. On solution, yes, solution is something that can be used for pediatric population. That is true. A lot of the fever medicines that we have, Tylenol, ibuprofen, they are available in solution form, but one of the biggest challenges is the stability. Do you have enough dose that you can get in a solution, and is that going to be stable enough? So sometimes because of stability issues or the form of the drug that you are developing, you need a solid oral formulation. And plus, solid oral formulations tend to be more stable and less susceptible to the risk of microbial microbial growth. That's another preference that folks have going to solid formulations, whether they're tablets or lipid or multi-particulates, over solutions. And then also think about the packaging and the label, not the labeling, but the packaging burden that a solution puts. You can have a few hundred milligrams of drug in 10, 15, 20 ml solution. So you have a much bigger volume for a small dose. When you have the solid form, you're shrinking that. So your packaging costs, your distribution costs, and everything, the weight of the medicine is low. So there are some logistical advantages to going with multi-particulates over solution as well.
Thank you for the answer. So we go further. Can we make nanocrystals of BCS class 3 drugs?
You can make nanocrystals of any drug. We have very interesting milling technologies. But in the context of multi-particulates, yes, there have been some cases where you have crystals or smaller crystals, milled crystals, or even nanocrystals that can be encapsulated or that can be basically added to your lipid multi-particulates, or you can also spray them on beads. It totally depends on the physicochemical properties of the drug and the release profile that you need and the patient population. But yes, there is precedence for that.
Thank you very much. And what are the key challenges and regulatory considerations in confirming pediatric drug products developed through multi-particulate technology?
It's a very good question. I am not a regulatory expert, but what I can say is that I'm just looking at the question. Yeah. When you're looking at challenges for any regulatory considerations, so I wear a CMC hat, chemistry manufacturing controls, which is the drug product, drug substance, the manufacturability. If you look at quality by design principles and ICH guidances on quality by design, those are the things that you have to consider. You have to have a dose that you have to identify certain target product profile attributes, whether it's your dissolution rate, whether it's your assay, water content, and you have to be able to show that these are repeatable, these are concise, and they're precise, and they are scalable. So sort of the same considerations that you have from a CMC perspective for tablets, you have to bake them in. And then the other consideration is there's some specification parameters that are unique, that can be unique to multi-particulates. You have to consider those as well, because you have smaller particles, higher surface area, different, a few different considerations, but yeah.
Thank you very much. The next question reads, how can we enhance permeability and determine it using simple method in lab?
That is a very interesting question. There's a lot of research on enhancing permeability specifically. Solubility can be enhanced by going to the amorphous form of the drug. Permeability enhancement is certainly something that is not easy to answer. And again, like any doctor or lawyer will tell you, they will say it depends. It depends on a lot of things. But for permeability, you can look at permeation enhancers. There's been a lot of research, especially with the bigger small molecules that are getting the PROTACs or even oral delivery of oligonucleotides. You're seeing the use of permeation enhancers that are needed. But sometimes if you don't need permeation enhancers, it's basically the other framework of thought here is that sometimes you have these colloidal species that are formed with drug polymer aggregates, colloidal species, and that helps shuttle the drug through the unstirred water layer. That could help in permeation, but it depends on the extent and the mechanism of permeation enhancement needed. Whether you need to physically have your epithelial cells, create pores in them for your drug to pass, or you just need that shuttling effect, which you can get through colloidal species. So there are different ways that you can undergo permeation enhancement.
Thank you very much. And the next question is, are there any specific expectations for taste masking in pediatric products using these formulations that developers should be mindful of?
Yes, so one of the things about taste masking is, depending on the patient population, there's precedence of certain excipients. So you have to, unless you want to go through the ordeal of approving a certain excipient, through the regulatory bodies. So there is a slim choice not, I would say slim. You have to be mindful of what's the precedented use of excipients for taste masking. The other thing is some of these particulates, such as lipid multi-particulates, have some inherent taste masking benefits. So it also depends on how bitter the API is. Is it moderately bitter? Is it too bitter? Can you get away with the coating, or you need some sweetener? So it also depends on the properties, of the API or the drug.
Thank you very much. And over the various forms of drug form administered for pediatric use, how preferred are the multi-particulate form in present scenario?
I think they're very preferred, especially over monoliths and tablets. There has been precedents, commercial precedents. You have seen big pharma and biotech invest and commercialize products that are multi-particulates. And now that they have done that, the regulatory CMC pathways for those and the CMC challenges and guidelines are present. So it's not that you're navigating, you're reinventing the wheel. No. So.
Okay, thank you very much. So those were the questions that we collected so far. Of course, if there are any further questions, you can always contact FIP Digital Events, and we will forward the questions to the speaker. And please also don't forget that the webinar has been recorded, so it is available on FIP Digital Events website. Now I will hand it over to Matthew for some final words.
Well, Dr. Arora, this has been one of the most interesting presentations we have had lately. I've been looking at the comments that your participants were making on your presentation to a person. They're very complimentary. So thank you very, very much. This has been a very interesting presentation. And thank you for bringing up this new idea to the wide world of pharmaceutical scientists around the world. We hope to see you again, Dr. Arora, sometime soon.
Julian, thank you so much for this opportunity. I really enjoyed giving this presentation, and thank you to everyone for your time attending. I know some of you are in time zones that it's not a convenient time, and you're sacrificing some family time to join this, so I really appreciate that. As Nicolina mentioned, if there are any questions, please follow up. We'll be happy to answer them.
Great. Thank you. Thank you, Jaspreet.
Bye-bye.